Professor Karim Adjou. I am from the Veterinary School of Alfort in Paris, in France. And this presentation is the work of my PhD student, Linda Sahrawi. She's now in my lab, and she's coming from the National Veterinary School of Algiers, Algeria. This presentation is entitled Occurrence and Genotyping of Cryptosporidium and Giardia in Calves in Algeria. As you know, Cryptosporidium and Giardia are very important in veterinary medicine. Cryptosporidium is one of the major agents of the neonatal diarrhea in young ruminants, such as calves, goats, and lambs. And Giardia duodenalis is one of the most frequent parasites of intestinal enteritis in human medicine. Let me show you some few data concerning ruminant breedings in Algeria. In Algeria, there is 2 million cattle, 5 million goats, and 27 million sheep. And these data come from the Ministry of Agriculture of Algeria. And this country is located between, it's located in North Africa, between Tunisia and Morocco. And the most farms, animal farms, or ruminants farms, are located in the north of the country because the other part of the country is the Sahara. Except 3 recent publications on cryptosporidiosis, there is no data available on Cryptosporidium and Giardia in small ruminants. So my presentation this morning in the small ruminant session was focused on lambs. And this afternoon, I will focus my presentation on calves. The main purpose of this PhD study is to characterize Cryptosporidium and Giardia isolate in calves from different regions of Algeria, but specifically in the north of the country. The first specific objective is to identify the presence of Cryptosporidium genus and Giardia duodenalis. Second, to identify C. parvum subtypes, and especially the zoonotic subtypes, because there is a zoonotic concern. This is a problem of public health for Cryptosporidiosis as well as for Giardia duodenalis. So 62 fecal samples were collected from different farms, from different part of the north of the country. And these samples come from young ruminants. The age was less than 3 months old. Most of the animals have diarrhea, and few of them were without diarrhea. The samples, fecal samples, were screened by immunofluorescence staining. We used Merifluor kits, Cryptosporidium and Giardia kits. They are available in Meridian Diagnostics Laboratory. And after that, the samples were screened by nested PCR for the small ribosomal subunit ribosomal RNA gene called PCR18S in order to identify the genus Cryptosporidium. And this is followed by the restriction fragment length polymorphism in order to, using 2 enzymes in order to identify the species. And when C. parvum species is identified, we performed subtyping with PCR and the sequence of a surface glycoprotein 60 kilodalton gene. This is for Cryptosporidium. The same protocol was applied in Giardia. The identification was first made by immunofluorescence, and after that, fecal samples were collected and were isolated, and DNA was extracted and PCR amplified by PCR using specific primers. And the sequencing here, analyses were carried out to determine assemblage groups, and especially zoonotic assemblage. Let me show you the results of this study. So from 62 fecal samples, only 40 were positive for immunofluorescence. And from this, which correspond to 58%, and this 40 samples were treated by 18S PCR, and we had 23 positive PCR on 40. And from this 23, we identified 19 C. parvum and 4 C. bovis. If we look to the age of the animals and the diarrheic status of the calves, you can see here in orange C. parvum results. All calves were diarrheic, and the most of them excreted the Cryptosporidium, and most of them have less than 4 weeks of age. The 4 calves aged more than 4 weeks old excreted a relatively small amount of oocysts. This is the data for C. parvum. If we look to the C. bovis, you can see here all animals were diarrheic, and they are all the animals have less than 4 weeks old. 3 calves excreted relatively fewer oocysts of Cryptosporidium. The next step was the subtyping, and as shown on this slide, the subtyping of the C. parvum give us these results. On 13 succeeded samples, we had two form of subtype families, 2A here, and 2D subtype family. And the predominance is of 2A subtype family. This is very important because I will show you after the comparison of our results with the previous data done in this country. For Giardia duodenalis genotypes, first of all, I would like to remember you the different assemblage we have in Giardia. The first assemblage is assemblage A. This is specific to humans and other mammals. You have assemblage B, which is specific to humans and primates. Assemblage C and D are found in dogs. Assemblage E is specific to hoofed livestock, like ruminants. Assemblage F, specific to cats. G, found in rodents. And assemblage H was observed and reported from seal and ghoul. And sometimes we have mixed assemblages, for example, A plus E. So you can see here on the left part of the slide, from the 15 positive tested PCR, we find 3 assemblage A on 15, 10 assemblage E on 15, and 2 mixed assemblage A plus E. So this is a very important result, but it means that assemblage A, which is zoonotic, is present in calves in this country. So the elements of the discussion are, first of all, we show as previous studies the dominant presence of C. parvum in ruminant farms. Calves are commonly infected before they are 3 weeks old. All these data are compatible with the literature data. The most excretory animals are aged less than 4 weeks. We have 4 calves in this study. They are very aged. They're old, 38, 40, 45, and 81 days old. If we compare our data to other studies, you can see here in this table, you have study 1 done by Ushen and collaborators in 2016 in north of Algeria. You have a second study from Baroudi in 2007. The samples come from the east of Algeria. The third study was done by Ben Houda and published in 2017, and our current study is here. If you look to the results, the most important result is the subtype of C. parvum. So we identified C. parvum as our colleagues, and we identified the subtype 2A as our colleagues too. But the most important thing is the presence for the first time in cattle in Algeria of 2-day subtype, which is zoonotic, as well as 2A subtype. C. bovis was also found in our studies, and no Ryan A and Anderson E were detected in our studies, but I think it's related to the age of the animals, because Anderson E is cryptosporidism found in adults. So in conclusion, our findings show that Cryptosporidium and Giardia infection in calves are common in this country. This study confirmed the occurrence of Cryptosporidium in cattle and reported the molecular description of Cryptosporidium and Giardia in calves. C. parvum is the dominant species in neonatal Cryptosporidiosis in young ruminants. And as I mentioned before, we have the 2 subtypes, family 2A and 2D, were zoonotic. So there is some consideration for the prevention of this zoonotic disease in this country. Our findings suggest that calves isolates contain zoonotic assemblage A, and in this case, calves could be a potential reservoir for zoonotic Giardia genotype A. And the next step of this work is to study the circulation and the molecular characterization of Cryptosporidium and Giardia in goats' kids, because we have already done the work in sheep. I would like to thank all my collaborators in France, other team in France too, and other collaborators from the Veterinary School of Algiers. Thank you very much for your attention.